My name is Kris Sankaran. I'm an assistant professor in the statistics department at UW-Madison. Today I'm going to be sharing a tutorial on batch effects. So this was a topic, I feel like it's something that comes up a lot in any real collaborative work, but it's not something that we often see presented in research talks. Like, I've been to a bunch of the CGSI talks and only a couple of them are mentioning batch effects, right? But it's something worth knowing. At one point, I didn't, maybe I refresh this. Okay, I have notes that I'd written for myself at one point that I was so interested in batch effects and normalization that I wrote a whole series of blog posts that I never posted, but I put them online on some PDF just for today. If you go to these slides, like after this talk, I'll make sure the link for those notes also appears here. Okay, so why batch effects? What are they? Why should we care? So about a year ago from today, there was a big retraction of a Nature paper. It had claimed to find a disease, a microbiome signature associated with different kinds of cancer. Okay, so the setup was they had looked at the TCGA data. There's a bunch of microbiome measurements associated with TCGA. They had said that you could come up with a diagnostic biomarker based on gut microbiome, and different kinds of cancer based on the gut microbiome. This is kind of an interesting case because there was no research misconduct, right? They didn't fake the data. And in fact, there had been several rounds of debate between different groups saying, is the signal real or not? And the same group had been publishing reanalysis of these data saying there is some signal. But it turns out that this disease signature was an artificial signature. It had come from a misuse of some batch effect correction methods. Right, so if you just used the standard pipeline, batch effect correction in microbiome data, this was creating a signal that wasn't actually there. So where do these kind of batch effects actually come from? Right, so in that case, it was a hospital-to-hospital difference. They knew TCGA, you get data from many different hospitals. They combine them all together. They knew that there's, if you made a PCA of the data just straight as you get it, the biggest effect you're going to see is a hospital effect. Right, that's what people mean when they say they see a batch effect in the data. It's not the really interesting biology that they want, it's just some hospital difference. And what's driving those sort of hospital differences, right, could be any of these kind of steps in the data preparation or sequencing. Okay. So even things like if you stored your samples in a -80 versus a -30, that has an effect on the kind of DNA that degrades is going to be different. If you use one extraction kit from one company versus another extraction kit, a different company, that can have a huge effect on the actual DNA that you retrieve. Okay. So all along the pipeline, each of these steps can introduce some systematic technical bias, right? And those sort of systematic technical biases, one hospital using one protocol, another hospital using another protocol, that leads to these big batch effects that you see in that first PCA plot. Okay, an analogy that I like is to think about this whole sequencing business as a very slow moving camera, right? It's there's the actual biology that you want to get at. So there's the counts of specific molecules, DNA molecules in some gut at some point, right? Or counts of specific bacteria, right? There's real biology going on in this biological sample, and then we're only getting at it after many, many steps, after many months, extracting data, collecting, finally getting sequencing, and then finally it's a spreadsheet, right? So it's like a camera, I think. There's the actual scene of this garden. We have all this glare, right? But we know the glare isn't actually what the scene is. We're able to easily tell what's the true scene and then what's some measurement noise. Kind of my idea, the analogy here is there's a similar kind of glare type effect going on in the abundance tables or expression tables that you get out at the end. It's harder for us to notice, right? Just looking at the spreadsheet, it's hard to tell oh, that's the technical effect. But it's there. a really nice discussion about this comes from Abhishek Sarkar and Mathew Stevens, who gave some talks earlier this week. They have a really nice discussion about the distinction between biological, like expression level modeling, and measurement noise and like measurement level modeling. So it's kind of helpful to keep these separated. Don't just look at the spreadsheet and assume that's the data, that's the truth, and we're going to just analyze that spreadsheet. Okay, so just reminding, biology is complicated. Think about what might be the reality before you got the final spreadsheet. Okay, so this is coming from one of the very first papers on batch effect correction. This is just sorting the samples by time, right? So this is from HapMap project. Just sorting it by time, there's some time points where expression levels look very different from the previous time points, right? Or like this is they're not time in like the true sampling measurement over time. It's like time in which you did the sequencing. Right? So the time when they sent this batch to the sequencing center, they got some systematic effects there. Okay, I'll just move through. General approaches, how you can deal with batch effects. Two big philosophies. One is you can try to correct for the effect. So apply some very generic batch effect correction method, subtract out what you think are the batch effects, and now you can use whatever downstream analysis pipeline you have in mind. That's one general strategy. Second strategy is whatever downstream analysis you have in mind, first think about how the batch effects might, basically build end-to-end one analysis strategy that accounts for the batch effects. Does that distinction make sense? It's like, the second one is like, for whatever problem you're trying to solve, define a model that includes a batch effect term somehow. First is decompose it into two separate parts. They both have their trade-off, or like advantages, disadvantages. Somehow this is the more elegant approach to like include batch effects in your entire analysis strategy, but it's also the more time-consuming approach. Because every new downstream analysis, you need to maybe have a new model. We're going to be focusing on correction methods, because I think this is probably the more general interest, and there are lots of methods out there, so I want to make sure you know how to navigate those. How you can compare between all the different proposals out there. One, think about what kinds of transformations are allowed. Right, so some batch effect correction methods are going to allow really complicated transformation from one batch to another. Others are going to restrict you to very simple transformations. So that's one axis in which you think about them. And then once you know the family of transformations, how does it actually select? Like how does it optimize across all those potential transformations? Right, so these are the two things you need to figure out before comparing two different methods. So just example, location scale transformation is like, if you had measured everything, just one batch, maybe this gene has this distribution, but then you measure using one extraction kit, there's a little bit of a bias, it maybe rescales things slightly. Right? So a batch effect correction method would be wanting to go from data that look like this across the two hospitals to data that look like this. I'm going to talk about two methods. Combat, this is like a very classical method, but it's very influential. It's also kind of the starting point for a lot of modern methods. And then I'll talk about CellANOVA. This is a more recent single cell specific batch effect correction method. I just wanted to highlight, what are the kinds of adaptations people have when they propose a new batch effect correction method. So combat. Combat stands for combining batches. Where does it come from? So the notation. We're going to use yi, gene expression across G genes for sample I, xi. These are known biological factors. So it could be like, which treatment group did it come from? Disease, healthy, maybe time points. M of I is going to be indexing which of the M batches the sample comes from. So like M hospitals. And this is the entire model. So there's some kind of global gene profile. There's some effect, depending on what group you're in. right? So you move in the direction W when you're in treatment, for example. And then this is like a location scale transformation. So each batch has some location shift, and you also can distort the noise, right? So it's like you can have a wider distribution, narrower distribution, depending on which batch you're from. Once you fit this model, you can subtract out B and kind of standardize by lambda. So you can kind of subtract out the batch effect to get the batch effect corrected version of Y. Okay, question?
Sorry, what's delta MG?
Or lambda?
Yeah.
This?
This. Yeah. Inside.
Oh, yeah. So this is like so this is going to end up being a hierarchical model.
Oh, okay.
Yeah. So there's going to be some prior on on these touches. Yeah, that's a good question. Yeah, and that's also important. Like this is diagonal. It's not like some arbitrary change in this G-dimensional covariance. It's just rescaling one gene at a time can have narrower or wider.
How do we define a batch? Like, would a person be a batch? Or the collection of people?
Okay, so in this, like a person would be one sample, and then the batch would be like a hospital. Also, an important point here is that it assumes that you know the batches, right? There are some around the same time. There were other methods like SVA that this doesn't assume that you know the batches in advance, right? Actually, the CellANOVA is not going to assume that you know the batches necessarily, right? So that's helpful if you have like the operator might have an effect, but you don't remember who ran which experiment. So that can happen. Any other questions about this model? This is a relatively straightforward model. I mean, it's just a linear model. Kind of the magic that allowed this to work when it was published is it's doing some sort of shrinkage, right? It's I kind of briefly mentioned it's a hierarchical model, so I'm not going to go into the detail how exactly they do this, but it's like they put some normal priors on B and on the variant, or like an inverse gamma on the variances, and they estimate the final B hat, lambda hat using empirical base. It's the entire thing. This picture is from their original paper. Each point is one gene. The circles are the mean, or the variance and the mean of that gene across the entire data set, estimated either without shrinkage or with shrinkage. And the point is, when they apply that hierarchical approach, like a lot of these very high variances are getting pulled towards the overall typical variance for most genes. It stabilizes thing, and this is what finally gets returned. Okay, so that's combat. Any questions besides that? Yeah.
Does that account for also across individuals, things like differential library depth or things like that?
No. This is just like batch effect correction, ignoring normalization more generally, right? Like these Ys are already standardized.
And W, is that like some loading matrix that gets learned?
W, it's like a coefficient matrix. Right, like if it were one gene, if this is just like one coordinate of this, then it would be like Xi transpose Wj. And then Xi is known, Wj has to be estimated. So it's like a matrix of coefficients across all the genes. Yeah. I should follow.
Maybe
Oh yeah.
Answer this later, but how does shrinking not cause biases?
Well, it does introduce biases, right? But then you hope that the improvement in variance is worth it, or like the stabilization of variance is worth it. But this is kind of also an important point that this is a method that's still somehow very popular, even though when it was introduced, there's typical sample size like 20, like 30. And this was really, really important then, and maybe it's not so important now. Yet we still continue using these tools, right? And also related to like the depth question, it's like nowadays, people would apply some sort of normalization here. This was originally for microarray data, right? So in the microarrays, you could do a log transformation and everything's very nicely modeled by log, right? People have come up with variation, like combat-seq is like a negative binomial version of this. but it's the same basic model and estimation. Okay. So, some of the subtleties when using batch effect correction methods. Like, important not to use these blindly. This paper had a very interesting experiment. They went through the combat-seq vignette, just the usual R markdown. You could run, get the output from their combat-seq. But instead of using the real data, they replaced the data with standard normal, just standard normal data. So there's no signal here, right? And then you go through the entire pipeline, you get the figures out at the end. This is the p-value distribution. There's a QQ plot of F statistics, right? So it looks like there's some signal here, right? So what's happening in this example is, I mentioned like if the treatment and control had been balanced across each of the batches, then this does not happen. Then you get perfectly uniform p-values. But there's an artifact that can be introduced when you have some imbalance in the treatment and the control within the batches, where when you now try to subtract out that batch effect, it introduces a treatment effect when there hadn't been any treatment effect. Right? So if like treatment and batch were orthogonal to each other, then this is okay. Then this will be uniform p-values. But if there's some correlation between the treatment status and the batch, that introduces this effect, even if there's no signal in the original data. So kind of one kind of paper to be aware of. Second kind of issue, this was kind of a cute paper that found kind of before you even try integrating data, you might want to run a test for whether it even makes sense to integrate the data. Right, so the cute picture they have is like, imagine these are like 3 cell types in hospital 1, hospital 2. Here it makes sense to apply some sort of integration method. You could imagine a location scale transformation that's going to align these 2 data. This one, it doesn't make any sense. Like these hospitals are measuring very different things. It doesn't even, like how can you even try to integrate this, right? So the argument is like, if you apply some of the methods, especially the very powerful methods, things more powerful than combat, they will be able to make it seem like the data are exactly overlapping afterward. But that's like an over-integration, is what they would call it. Okay, and then back to this microbiome paper. I'm actually going to go into a bit more detail about why this one fails, right? But before explaining that example in more depth, like, okay, I just highlighted a few problems that can happen when you use batch effect correction incorrectly. So what are practically can you do so that you avoid these sort of minefield, right? This is informal. I don't know if this is like been studied really like, you know, quantitatively or anything, but my feeling of the good sanity checks, one is I try to make sure that I can get the effect on the original data, like one batch of the original data, and then compare it with the corrected on the combined data. If I'm noticing like totally different genes, totally different species on the like largest batch versus corrected data, that's kind of fishy, right? So that's one check you can always do. Make sure that there's some consistency across batches or like corrected and individual batch. A second thing, simulation. Right, so the example I'm going to show, and even like this toy example with the p-value distribution, those were just generating some data, applying the pipeline, making sure that you get reasonable results on the simulated data. So this has been kind of pretty much all tutorial. I'm not really going to talk about my own research in this tutorial, but this reference is my reference. So this is me, like we wrote a little discussion on like how you can use simulation to sanity check some microbiome analysis, and batch integration is one of these. Okay, so how did this, how can you understand what happened in that nature retraction using simulation? This has come from Jerry Tonkin-Hill at Melbourne. This is just a mixture of Gaussians simulation, so it's really far from any real microbiome data, but it's enough to get a sense of what might have happened. So imagine these 4 groups are like 4 hospitals. The 2 colors are like disease, healthy. Okay, so here there's no signal. Each hospital, there's no disease healthy group effect. You apply the supervised batch effect correction method, And it does what you expect. Like the 4 hospitals are now exactly overlapping. There's no systematic structure across the diseased and healthy. This is what you want. Now simulate it where there's this imbalance, right? So now there's like no disease in this one hospital. There's like a really heavy skew in the other hospitals. And now if you correct, it introduces some effect, right? And this imbalance is not that implausible, actually. Like if you think about TCGA, some hospitals specialize in some kinds of cancer. So you get a lot more of those cancers in those hospitals compared to every other hospital, right? So by trying to remove the hospital effect, they were accidentally introducing a cancer effect. That's what allowed them to seem to be able to predict the difference between the cancers. This is kind of unfortunate because there might actually be some diagnostic potential from microbiome and cancer, right? But then the fact that you could run the simulation and see like this sort of effect means at least that analysis you can't rely on to get that conclusion. Okay. Last few minutes, I just want to highlight this CellANOVA idea. It has one really nice insight, which is instead of just thinking of batch effect correction as something you do after you already have you already just gathered the data, think about when you're designing the experiment, how you might have designed it in a way that makes batch effect correction easy. Okay. So, replication and controls, these are kind of recurring themes in statistics. They're really focusing on like define the controls so that you can estimate the batch effects based on those controls. Yeah, just skip this. So the notation, I mentioned this is a single cell method, right? So now each person is like a Y1 through Ym. These are now different people. And each of those is a matrix. So each person has many cells, right? So many cells, many genes for each person. The key thing that this method requires that's like not in combat and not in kind of most batch effect correction methods is it's going to assume that you have a subset of the samples that they'll call like a pool of controls, that these are going to force to overlap one another, right? So any systematic variation across these, it's going to remove. And then they're going to use that as the basis of the batch effect correction. The intuition I have for this is like if you had the exact same biological sample and you had done many, many technical replicates of that one sample, you look for the variation among those replicates for that one sample. Any systematic difference you notice among those replicates, you're going to subtract that out. And now apply that transformation to all of your data. Right, so use those pool of control, find the systematic variation, remove it, now apply that same transformation everywhere. This is kind of how they formalize that intuition. It's a little bit messy. CM, so this is single cell data, right? So the cell, each row might be coming from a different cell type. So think of CM as like encoding which cell type it comes from. Global effect, there's going to be a distinction between batch variation and biological variation, right? So there's like low rank matrices for both the batch effects and for the biological effects. Unlike combat, this X is unknown, right? They're not assuming that you know which kind of true biological subspace, or like what's causing that true biological subspace. Right, so just starting with this formula, if you just have two low rank matrices added to each other, if you didn't have any experimental design structure, this is not identifiable. Like you can't distinguish between those two low rank, like just adding those together. But the main idea is going to be that since we know like that the pool of controls will have no biological variation, you use that to estimate the batch low rank part. Okay, this is where I try to draw a picture and see if this somehow makes sense. So imagine like this is one cell type, and this is like batch 1, batch 2. And I mean, this is like a gene 1, gene 2. So it's like the centroid for this cell type in the two batches. And maybe there's another cell type like out here. Right? So here the batch effect is shifting the centroids in this direction, right? But there's still these two different cell types. How do you actually try to estimate the direction V? Okay, R is the centroid matrix, right? Like it's with all the pieces together. It's going to subtract out the average among all the controls. So like these are just the controls. The difference we're seeing in the white and the black, this is just the batch effect, right? So these are like my R bar first row, R bar second row. Once I do this subtraction here, I have like basically overlapping, doing this right. Okay, yeah. And now you just look for the variation that remains. And that subspace that remains, this is the batch effect subspace V. Okay. So that's the main idea. Once you have that subspace V, you project that out, and then you do the SVD of the projected out version. That's the biological subspace. Okay. I kind of rush out over some of the math detail, but the main point is like you can use that pool of controls. That experimental design helps you with the batch effect correction. And just to close, just putting these side by side and showing, even though combat's like 20 years old, the basic algebraic structure is pretty similar actually. So still worth knowing these classical papers. Okay, so yeah, hope you have a little bit of sense of like why we should care about batch effect correction, some of the basic methods, and also like some checks you can do. So it's not just a matter of applying some method and trusting it works. So happy to take any questions. Thank you.